All right, so these notes are going to require a lot more writing. And one year I actually said, why am I making them write it all? I'll just give them the notes and just almost like on a PowerPoint sort of thing. Like I think Jill Birdie does that sometimes. But I found when students didn't take the time to write it down themselves, they actually didn't learn it as well. So, okay, so I want to start the way the video did. I want to talk about an observational study. Okay, think about what it's called, observational. All they do is observe, okay? And so they were doing the coral reef example, and that you still have to have certain outcomes that you measure. They said they talked about water quality, they talked about the number of large fish, and whatever was growing on the coral reefs themselves and things like that. So they had things they were measuring, but they didn't do anything but look and measure at what was there. They didn't impose a treatment. They didn't say, well, let's add this chemical to the water and see if that gets rid of some of the pollution. They didn't do anything, okay? No attempt is made to affect the outcome, okay? So this example says, does living near power lines increase the risk of leukemia in children? Do you know what, like power lines, we talk about the electrical thing. Can you picture, do you know what an Edison corridor is? Okay, so do you know what Detroit Edison is, the electrical company? And there are certain places that there'll be like a whole long strip, miles and miles of the big metal structures that almost look like little mini Eiffel Towers, okay? There are some people who feel like if they live near those Edison corridors, that's bad, that you're getting all those electrical currents that are near your house and things like that, okay? So there are some people who believe that. So the National Cancer Institute, the NCI, compared 638 children who were diagnosed with leukemia before the age of 15, and they compared them with 640 children who did not have cancer. The researchers measured magnetic fields in various rooms of their homes, at the front door. They recorded information about power lines near the homes and the homes where the mothers might have lived during the pregnancies themselves, okay? All they did was measure what was there, okay? So explain why this is an observational study and not an experiment. No treatment was imposed or given. Okay, they didn't do anything. They weren't trying to see if a certain type of medicine might help, put the cancer in remission. No, they weren't doing that. They were just observing and measuring, okay? Explanatory and response. Now, explanatory, remember, this is the word you might be more accustomed to, the independent variable. Response, you might be more accustomed to the dependent variable. But bottom line is explanatory is X, response is Y. And we just use different words in statistics because when we study probability, independent and dependent has a different meaning. So what you have to think about here is what are we thinking what might be causing, and I'm using that word generously because we can't talk about cause and effect here, and what might be the response. So what they might think is happening, they might think that the power lines might be causing a response of the leukemia, the cancer. Now I'm going to emphasize again, I am loosely using the word cause there. You cannot establish cause and effect if it's just an observational study, okay? And that brings us to the lurking variable. A lurking variable sounds like a weird phrase, but I think I've used it with you guys before. A lurking variable is something else that actually might be the cause. What could be the cause of cancer? Okay, it literally could be genetics. Unfortunately, there are some people who might be predisposed to getting cancer. Could it just be a random gene mutation? Could be. What else might cause cancer? There are so many things that could be happening in these homes that we don't know, okay? So maybe it's habits, which could be smoking, drinking, whatever, okay? A couple years ago, probably more than a couple years ago, you guys know what like a nonstick pan is? Yeah. you guys might have been too young to remember, but at one point there was a thing that they were concerned that the non-stick coating, that when it heated up in the pan was releasing carcinogens. Okay, well that would be kind of bad because now we're all eating the food that was cooked in the pan. Okay, so I'll just say other environmental elements. There are so many other things that might be the cause. All an observational study does is give us a relationship or an association. We can't say that there's a cause and effect. Yeah, we talked about looking there. Remember the fire example? All the firemen show up at the fire? Okay. So a lot of people then will say, well, then why do we bother with observational studies if we can't get a cause and effect? The video mentioned that. The benefit of an observational study is you're not impacting the environment. You're observing what's there and you're not damaging further what might be there. So an observational study allows a researcher to look at what's happening without causing potential further damage. Okay, so there is some benefit to observational studies. So now I'm going to back up. And again, the only reason I did it like this is these two examples, which were about experiments, fit better on this page. Okay, so in an experiment, some sort of treatment is imposed. In the video, they had new medication 1, new medication 2. They did a combination of the two meds. Then they had the active placebo, the old medication, and the actual placebo, okay? So you actually impose some sort of treatment. A study published in the New England Journal of Medicine compared 2 medicines to treat head lice: an oral medication called ivermectin and a topical lotion called, and I'm not really sure, I pronounce this malathion, but I'm not sure it's right. I don't know for sure, but we'll go with that. So researchers studied 812 people in 376 households in 7 areas around the world. Of the 185 households that got randomly assigned to the ivermectin, 171 were free from head lice. That sounds pretty good. What is that? 171 out of 185. So 92% were lice-free. That sounds pretty good. Okay, compared with only 151 out of 191 in the other. That's only 79%. Now, I'm going to kind of jump ahead to a different chapter, but eventually what we're going to learn how to do is, when you look at those numbers, would you feel like ivermectin is better? It sure seems like it, okay? We are going to learn how to decide, how to determine, statistically speaking, is that statistically significantly better? It clearly is better, but is it so much better that we would say, I think that the medication was the reason, not just some people, the lice just died off, okay? So we'll talk about that in a later chapter, but for right now, why is this an experiment? Okay, there were treatments. Okay, there were 2 medications. Okay, there were actual treatments. They didn't just observe the households and watch to see if the lice died off. They said, you try this medicine, you try this medicine, and they compared the results. Okay, the experimental units. Now be careful on this. The experimental units, you're going to be tempted to say it was the 812 people, but it wasn't. It was the 376 households, okay, the 376 households, because not only was it person by person getting the medicine, they assigned an entire household to one of the 2 meds. So it might be 5 people took that med. It might be 9 people, okay, you don't know. It was the entire household. The explanatory variable, we think that the medications will be the reason that the response will be, yeah, I'll say that we're hoping we're going to be lice-free, okay? You might say the amount of lice, I don't know, but the bottom line is we're going to say that we think the amount of infection from lice or whatever it is, the amount of infestation, I should say, depends on what med did we take. So you can still think of it using your old independent, dependent words if you want. All right, the treatments here are the two meds. So it was either the ivermectin or the malathion. And confounding variables, which by the way, there's a typo. There's no U in the beginning of confounding, C-O-N. A confounding variable versus a lurking variable. You can almost use them interchangeably. I have looked this up numerous times and I get contradictory information, but what I get most often is they call it a lurking variable in an observational study. They call it a confounding variable when you're in an experiment. But I see them used interchangeably all the time. Some people say they're all lurking variables and they all result in something called confounding. And the word confounding kind of sounds like confusion because that's what happens. A confounding variable is something that is happening that it's going to confuse my results. So what could be happening in these households besides the meds that could be impacting the amount of lice infestation?
Well, like if somebody has like a more severe case, then like the meds wouldn't be able to.
Oh, so possibly, I don't know much about it, but we'll add that, okay? The level of infection or severity of infestation. I don't think it's really considered an infection when you have lice. Logan, you had your hand up. The climate? I don't know either, but maybe you're right. Maybe because we're talking inside the house. Maybe some people keep their how do you guys keep your house fairly warm or cold? Cold. Okay. Yeah, my mom always did too. She liked it really cold. Okay, saved money on the heating. But yeah, we'll say climate. You're right. Maybe if the inside of the house, or maybe just spending more time outside. Yeah. Okay, I'm with you. We don't know. That was my first thing, was the type of shampoo or conditioner you might use. Or do you use a gel? Do you use a mousse? Do you use a hairspray? You know what I mean? Hair products. I would think that, and not only hair products, but I remember, did they do lice checks when you guys were in elementary school? They had these big pointers, almost like knitting needles, and they pull your hair apart, they're looking at your scalp, okay? So, but it's not even just on your head. Lice could be anywhere on your body, okay? You guys know, during World War II, that's why they, the prisoners, they would make them shave their whole bodies. That was why, because it reduced the chance of lice infestation in the prisons and things like that, okay? So all of a sudden, hair products or even soaps, things like that. Yeah, what?
Well, if it's an oral medication, maybe it's consistency, you're actually taking it when you're supposed to.
Okay, taking when you're supposed to, or what if you took it, what, I know sometimes when I take a liquid medication, I feel like if I drink something right after, because I hate the, does that affect it? I don't know.
Does that qualify as a confounding variable?
Well, but it could.
Wouldn't that have to do more with the experimentee?
All right, but how the individual, how they accept their treatment certainly could now be a confounding variable, okay? Because if they take their medication improperly, so now you think, oh my gosh, that med's not working, and maybe the med was going to work, but they took their medication improperly, okay? Maybe they're supposed to take it with food, okay? We could make this super long list, but you're getting the idea. The idea is, if we want to establish cause and effect, if we want to say the amount of lice, was this 92%, reduction? Was this due to the ivermectin, or was it due to you washed your hair with a shampoo that killed the lice? Okay. This is what we talk about. We talk about controlling our environment. If you really want to make a good scientific experiment, you have to control as much of this as you can. You might say, okay, I need all of you to use this shampoo, okay, so that I can make sure that that's not really what's killing the lice. I want to make sure that you're all taking the meds at the same time of day. You're going to take it right when you wake up, before you eat. So you can now come back and say, okay, I can't blame it on any of that other stuff because they all did it the same, okay? Now, you might say, how am I supposed to control all of that? And the answer is the researcher can't always control all of that. Okay, he could say, I want you to take your medication at 9 o'clock every morning on an empty stomach. Does he know everyone's doing that? No. Okay, so a researcher also has to have a way to, if I can't keep this under control, what can I now do to fix that? And we'll talk more about that coming up. But a researcher should try to control as much of this as they can. Okay? In the bad video, okay, in the bad video, the whole thing about, you're trying to ask someone how good you feel. Allowing the one person to sit down and rest and making the other person stand the whole time for an entire hour, okay, that's a confounding variable now. Maybe the second person felt worse because now they're exhausted. You made them stand there for an hour, okay? So that would have been easily controlled. Make them both stand or get another chair. Make them both sit. All right, some experiments studied the joint effects of several explanatory variables, and then the vocab gets a little bit murky, okay? And I'm going to tell you, this is one place on vocab I wouldn't, confounding versus lurking, don't stress too much. They're used interchangeably a lot. When we talk about treatments, there are factors of treatments, there are levels of treatments. Don't get too hung up on which is the treatment, what is the factor, what is the level. But when they start using these words together, it means there's probably a combination of things going on, okay? So in this case, does adding fertilizer affect productivity of tomato plants? How about the amount of water given to the plants? To answer these questions, a gardener plants 24 similar tomato plants in identical pots in his greenhouse. I want you to think about that right there. That was the gardener right there controlling some potentially confounding elements. We're not going to just get 24 plants. Some of them are only this big. Some of them are already this big. Not different breeds. They're all going to be similar tomato plants. They're all going to be about the same size. They're all going to be about the same kind. Okay? Like my son was out last night shopping. They needed potatoes for a lab. And his coworker said they need to be russet potatoes, not Yukons. Not the red skin. Think about that. Do you know what the Yukon Gold are? The yellow outsides, the red skin potatoes, and a russet potato is like a normal brown. Okay? Do you understand that doing a lab with different types of potatoes could cause different results? So his coworker says they need to be russet potatoes. Well, this guy said, okay, I have similar plants and they're going to be in identical pots. Okay? If I take a plant and I put it in a tiny pot like this, now all of a sudden the roots have no room to grow. If I put it in a really big pot, now when I dump a little bit of water in, now the saturation of the soil is going to be different. So similar plants, identical pots, they're all in the greenhouse. Not one sitting on the east side of the house, the west side of the house, in the garage, in the basement, okay? All in the greenhouse. So all of that deals with eliminating your potentially confounding variables. You try to make the environment as similar as possible because now he's going to try to decide what is the best way to grow my tomatoes. He adds fertilizer to the soil in half the pots. So he's going to compare fertilizer versus none. And then he waters 8 of the plants with a half gallon of water. 8 of the plants get a full gallon of water, and the remaining 8 plants get a gallon and a half. At the end of the 3 months, he records the total weight of tomatoes produced. All right, and that's why I said you always have something you have to measure at the very end. Okay, the experimental units, I think that one's fairly obvious. Those are my 24 plants. Explanatory and response. I think the weight of my tomatoes will depend on how much fertilizer and how much water. So I have 2 explanatory variables here. But now we start talking fertilizer, H2O, weight of tomatoes, if you can't read that. But the issue is, this is where we get into factors and levels, okay? And I would say, in general, I think people will probably use treatments and factors somewhat interchangeably. Levels actually seems to kind of make sense. Okay, the level is how much. So one of the easiest ways to show this, I make a tree diagram. All right, if there are 24 plants, the first thing he does is he said, half of them, fertilizer, no fertilizer. Okay, but then out of these 12, I have 3 different levels of water. So I'm going to have a half gallon, 1 gallon, 1.5. So this is my H2O. And over here, half, 1, 1.5. And so there would be 4 in each of these. And so that matches up with what it says in the description. 8 of them are getting a half gallon, 8 are getting a gallon, 8 are getting a gallon and a half. So really, how many treatments are there? There are actually 6. There's fertilizer, half gallon, fertilizer, 1 gallon, fertilizer, 1.5. No fertilizer, half, no fertilizer, 1, no fertilizer, 1.5. There's actually 6 treatments going on there. So we would say the treatments are all the combinations. And now I would say the factors are the fertilizer and the water, and the levels is yes or no, and how much, okay? To get a good scientific experiment, it can get complicated trying to come up with that process, okay? All right, so now let's jump up here and continue talking about experiments and what makes a good experiment. You want to have a comparative experiment. Use 2 or more treatments so that you can compare the results. Okay, I use the example of my mother. She has high blood pressure. She also has had quadruple bypass, so she has, a weak heart. Okay, so let's say for her high cholesterol, the doctor says, you know what, Miss Eschenberg, I want you to try this new med. If the doctor just gives my mother this new med and her cholesterol goes down, do we know that med did it? We got nothing to compare it to. Okay, now if that doctor had a whole bunch of people who were similar to my mother and gave them all the med, and a whole bunch of them had their cholesterol go down, now we can start saying, okay, maybe it was the med. But we don't even know that for sure. We have to look at the entire experiment, okay? You want to be able to compare it, okay? Now, everything I read over and over again says that a control group is not a requirement. But almost everything I read says it makes your experiment better, okay? What a control group does is it provides a baseline to help determine if results were truly due to the treatment as opposed to, was it due to just chance? Maybe my mom's cholesterol just happened to go down. Was it due to a placebo effect? Do you guys know what the placebo effect is?
Yeah.
How many of you have AP Psych? Yeah, you're going to get some duplication on this also. So the placebo effect is a real thing, that some people receive a placebo. They get a med, it looks just like the real thing. They don't realize there's no medicine involved, but somehow they feel better just because there's something about the mentality. It's a psychological thing that they start feeling better because they believed they were taking medication. Or the video even said that just being under a doctor's care sometimes makes people feel better, okay?
I also say like confounding variables to be like
Actually, the confounding variables is going to be more. We're going to bring that more in here. Okay, but yes, it is related. And that's why I said most things say you don't need a control group, but it makes it better. Because yes, a lot of this is going to come back to the confounding variables. Okay, now the confounding variables, these three things right here in your book, they call these principles of experimental design. These are the three things that even on College Board, AP Central, these are the three things you want to talk about: control, random assignment, and replication. All right. Now, these two controls, they're not the same thing. You guys know what a control group is, probably from just science labs, right?
Yeah.
All right. This is a verb. This one is a verb. You have to take control of your environment. Okay, this is you controlling the environment to try to reduce those confounding variables. Control your environment. You want to reduce any potential confounding variables here. This is the gardener. He reduced his confounding variables by, I'm going to start with similar tomato plants. So we can't blame it on, we can't say that that plant had better tomatoes because it was a better plant to begin with. He reduced his confounding variables by saying, I am going to put them all in the greenhouse. So I can't say that this plant did better because it got the morning sun on the east, and this plant didn't do as well because it only got the afternoon sun on the west. He controlled his environment by saying, I'm going to put them all in the same pot. Okay? So you take control of whatever you can. Okay? No, I don't need to say that again. All right, good enough. Good enough. All right, random assignment. Now, here's the thing. As a researcher, you can do the best job you can. All right, back to the example with the lice that I said, how do we know that someone is taking their med at the right time, in the right manner? You gave them a special shampoo to use, and they're like, that stinks, and they use their own, okay? What happens now is, this is why we randomly assign. We use random assignment to put the individuals in their treatment groups to create somewhat balanced groups. Now, when I say somewhat balanced, what you're trying to do is you're trying to create similar groups. So basically what happens is you're like, okay, as a researcher, I controlled everything I thought I could, okay? But now you say, but what am I going to do about those people who don't take the meds like they were supposed to? They're using the wrong shampoo. Well, what you do then is if you have, and I'm making up the number, let's say you have 100 people and you want 50 to have malathion and 50 to have ivermectin, okay? You could number those people 1 to 100, and you could sit there with your random number generator, and you can say, okay, I am going to randomly generate from 1 to 100, 50 people. And I do it. Okay, person number 57, you're in this group. Person number 64, you're in that group. So you randomly assign 50 here and 50 there. And then what happens is all those people who aren't following the rules, we hope that they get mixed up a little bit. So you don't have all the non-rule followers in the malathion group and all the rule followers in the ivermectin group. Because now all of a sudden, if all the rule followers are here, maybe just because they took the meds. Maybe these people didn't get better because they didn't even take their meds. But if you randomly assign them, so you got some good people, some bad people, some good, some bad. Now we can still say, okay, now let's see if we can look at and see which med did better. The whole idea is if I can't control something, if I randomly assign them, you'll all get mixed up, okay? And then finally, replication, large N size. Have a large N size to ensure results are due to the treatment, or to help ensure all of this is to help. We can't really ensure anything. Okay, back to the example with my mom. Okay, if you only give the med to one person, okay, let's say my mom takes a new cholesterol med and her cholesterol goes down. We can't say the medication did that. Maybe my mom finally decided to stop eating the cheese balls out of the canister, okay? Maybe maybe she finally decided to exercise a little bit more, okay? So if you have enough people and enough people take that med and a whole bunch of them, their cholesterol went down, now we have a better chance of saying, okay, maybe it was due to the medication, okay? Some of them started walking more, some of them stopped eating the cheese balls, okay, whatever, but if you have enough people, you have a better chance of attributing it to your treatment, okay? All right, so that's as far as we got last hour. Also, I know you guys have your test next hour, so.
Oh, I'm locked in.
All right, speaking of homework.